For those of you who don't know, my name's Kevin Lang. I help organize the RIDE series here for the college in partnership with the research office. And I just wanted to start off with a few housekeeping notes here. So thank you for joining us here for our invited speaker talk today. Next month, we have two of our own College of Vet Med researchers, Dr. Amy Treefel and Dr. Emily Coffey. So please join us next month, June 11th. And then don't forget to also join us for the RIDE Summit, which will be happening on the second Wednesday of July, I believe. Don't quote me on the date, but keep an eye out for notes about that. I'm going to keep it really quick. I just wanted to remind everybody, just in the room, just please hold your questions until the end of the talk. We'll have plenty of time for questions then. I'll also be monitoring Zoom for those of you who are joining us virtually. Please type your questions into the chat and I will do my best to ask them here. So with that, I'm going to turn it over to Dr. Wells so that he can introduce our speaker for today.
Great. Well, thanks everybody for coming. Well, real treat today. Our speaker for this RIDE seminar, Dr. Jason Lombard. I've known Jason a long time. He's a veterinarian and epidemiologist and focused on dairy cattle for his career. He spent time in private practice after vet school and then had worked with USDA APHIS for, I think he told me 20 years, quite a long time, while at the Centers for Epi and Animal Health in Fort Collins. And a couple of years ago, he moved over to Colorado State University. Based on his expertise, particularly when this influenza situation started up, APHIS said, Jason, here's some money, help us answer some really important questions. And amongst that, he's coordinated some of his own projects, and he's coordinating and collecting data and supporting people in different states, including at Minnesota. So it's a really great opportunity to find out sort of on the ground from an epidemiologist, what's happening with this virus in dairy cattle? What do we know about how are these herds getting infected? What do we know about how it's moving around? And he's here for a couple of days and he's met with several of you already. And he'll be here till tomorrow. So anyway, with that, I'm going to turn over. Jason, what's new?
Thank you, Scott. Thanks for the invitation. Thanks for having me here today. My goal of this is really, if you have more questions when you leave, then I've succeeded. Because it seems like every day we have more questions that go unanswered than go answered. So I've only got about 120 slides. I think we can get through them in the next hour. So as Scott said, I did used to work with USDA, and they kind of said I needed to put this slide in all my presentations. So I'm not representing USDA, and part of the data that you'll see today was gathered through USDA funding. So I want to start out with acknowledgements because, man, it takes a lot of people to do some of the work that we've been able to accomplish. And essentially, USDA did come and say, here's for me a large amount of money to work with, and we want you to answer a lot of these questions. And I just moved to CSU 6 months prior, and there was a team of me. And I'm like, how eventually we got a lot of people on our team, and I think we're now where we can start answering some of the questions with a lot of the data that we've gathered. But we still have samples that were collected last July and August that haven't completely been tested yet. So we still have that little bit of backlog going on. But working with a lot of different folks and appreciate all their help. This is a research team at CSU that's kind of the core group. And the interesting thing, except for a couple of these, I guess Kathy and Madison, all the rest of these folks are previous USDA, not USDA personnel, but had connections to the National Animal Health Monitoring System where I worked for about 20 years. So I've kind of taken the people like Bruce and Lindsay that went into retirement. I said, you need to come out of retirement. You need to help me. And I, unfortunately for USDA going through some of the cuts, they just had some people go through the DRP. And I actually was able to snag one of those people. I just have to fund her differently because I can't pay her with this, because technically she's still be paying by the USDA through the end of her DRP. So just a little background on things I wanted to talk about today. Just a little bit on HPAI. And I'd worked quite a bit with HiPath in terms of outbreak response and doing epi in multiple states since 2022. But when it got to cattle, I think everybody's like, well, this is interesting. Well, we've got a lot to understand. We'll talk a little bit about disease spread. Early detection of infection in herds was a study we conducted with some partners in California. Shedding and serologic response, most of those herds were in Colorado, but we did enroll some herds in California as well. And then evaluation of comorbidity, BVD, BLV, and Yonis. That's one slide at the end. I'll just tell you that we have found no association to this point with any of those three diseases and shedding and of HiPath in cattle. But maybe we need to broaden what diseases we're looking at. So for those of you that aren't familiar with influenza, there's 4 types. Influenza A, which is usually associated with seasonal allergy or seasonal disease, which is what we typically get vaccinated for, but also global epidemics of influenza A. Influenza B has also caused some of the seasonal flu. C is not really seen very much from what I understand. And then influenza D is what we primarily find in cattle. So most of the time we think, well, we'd be talking about influenza D today, but unfortunately we're talking about influenza A in cattle, and really based on 2 surface proteins, the hemagglutinin and the neuraminidase. And so what we've got is in the current outbreak, and that's the H5N1 clade 2.3.4.4b, genotype B313. That was the cattle genotype. So we think this spilled over sometime in late 2023 through a single event into cattle and then spread through cattle. And now every state that's been affected, we know has been through cattle movements. Until we got to December of this year, and then in November, December of this year, and then I think in February, it was confirmed through USDA that now we had the genotype D1.1 had been detected in Arizona, potentially two different spillovers in Nevada. So a total of 3 potential now spillovers from wild birds into dairy. So what was, we thought once in a lifetime, the B313 into dairy now has been potentially 4 times in less than a year. This is the map, the USDA map from a few days, just a couple days ago. Total outbreak over 1,053 herds in the 17 states. This is the herds in the last 3 months, or last 30 days, I'm sorry. 3 states in the last 30 days. Idaho's really leading the list right now with over 30 herds, and that's based on the national milk testing strategy where they're going in and testing milk from either silos or bulk tanks on farms. And what they're finding in Arizona and Idaho is herds that have it in the bulk tank that the cows aren't necessarily showing any clinical signs. So is the disease kind of coming to a truce with the cows, and now they're not making them as sick, but we're still finding it in the bulk tank. So we've got some work to do. California kind of has burned through most of their herds already, and they don't have very many unaffected herds left, at least in the Central Valley part of California. Idaho didn't have a lot of herds that reported back last summer when it was kind of going through, but what we heard from the ground was that almost every herd in Idaho was infected. And that's true for most cases. I think only Colorado and California, up until the national milk testing strategy started, could say they had a good idea on the number of herds infected. But I can tell you in Colorado, there were 22 herds that were bulk tank negative. They started testing in July and talking to practitioners like, yeah, they were negative, but they were infected before the bulk tank testing started. So probably most of the herds in Colorado were infected as well. So just some significant dates. February 8th, 2022 is when we first saw HiPath in a commercial turkey flock in Indiana for this outbreak. And then in early 2024, this uncharacterized cattle morbidity event that was happening in the Southwest United States, primarily Texas and Kansas, New Mexico. And there were reports of large bird die-offs associated with that. A lot of cats were dying on farms. And it took them a while to kind of figure out, well, what's going on? And then finally on March 25th, we confirmed HiPath AI, B3.13 genotype. About a month later, USDA enforces a federal order that essentially required testing of lactating cows prior to interstate movement, because that's all USDA can do. Is they can't go in and dictate what a state does, but they can dictate what happens once the cattle leave a state. So interstate movement, they could require that. And then the other thing that was happening a lot was private laboratories doing PCR testing for high path and not reporting it to anyone. And so the federal order required now every laboratory that was running this to report it to the authorities. And then on February 2nd, the genotype D1.1 confirmed in Nevada and in Arizona. So this is a map that shows which other wild and captive wild mammals have been detected with high path. And you can see there's a lot of different species in there and in a lot of different states. So everybody asks, well, what about beef cattle? Why haven't we found this in beef cattle yet? We've looked in beef cattle and we've had some results that changed when we went back and retested, but to date we have not found serologic evidence of this in beef cattle, and we've looked. But it doesn't make any sense that we've got it in over 100 different mammals that beef cattle would be different from dairy cattle in terms of their potential risk of disease. So the current challenge is, how is the virus moving herd to herd? This is an old list from probably a year ago, and we don't have answers to these yet, really. How is the virus moving cow to cow? I think we have some additional information that gives us insight into maybe how it can circulate in a herd and what's going on in terms of pathogenesis, but it's, I think, really early, and we need a little bit more data to start confirming some of our suspicions. Are younger cattle and non-lactating cows involved in disease spread? We know that we have essentially nulliparous heifers that are aborting fetuses that are PCR positive. So we know that they're getting exposed. It's not through the milk. It's probably through oral or respiratory. How they're getting the virus delivered to them, we don't really know. And then, is reinfection or relapse already occurring on farms? I think that's sometimes really hard to distinguish, because a lot of times when we get out a long way in a herd, the CT values are so high that we can't do whole genome sequencing on the amount of virus that we're detecting. I think that the Minnesota herd that recently was worked up, that says that really is just probably a persistently infected herd. But I've heard in Idaho that they are having a different B313 genotype variant that's being seen in some of those herds that were infected last summer. So I think we have persistent infection going on in some herds. We have reinfection going on in some herds, but it's hard to document that because of sometimes the amount of virus that we have available isn't enough to do sequencing. So disease spread risks, known risks, lactating cattle. Every state that's been infected has been through the movement of infected cattle. A lot of times it was not known. So when the first cattle moved out of Texas, that herd that they moved out of was detected about a week after those cattle were shipped. But lactating cattle gets it to a state. Once it gets to a state, then I think there's a lot of other factors at play that dictate movement of the virus. People, this was kind of demonstrated when this first started in Michigan. I got called to go help with the strike team to figure out how it got from dairy to poultry. And what we found out was that there were people that were working at the dairy on the weekends from the poultry plant, and 2 months, 6 months before that, we wouldn't have necessarily seen that as a risk. But there were 22 people that were working at the dairy and then going back to the poultry plant. So we assumed that those people were bringing it back to the poultry. The assumed risks we have: service personnel and vehicle movements. There's a lot of those on dairy operations every day, and so they have to be considered somewhat of a risk. We assume feces is a low risk. They did a lot of PCR on feces initially, and we found one sample that was positive, and NVSL, National Veterinary Services Laboratory, said, quit testing feces. We don't think it's worthwhile to test. And we're like, okay, that's fine. Manure, which is a combination of feces and urine, though, could be a risk because we do find it in urine, not frequently, but we do find it. Almost every farm we find a cow that has it in urine. So then we start worrying about, well, what about lagoons and spreading of manure? And we've heard multiple stories about, well, the poultry farm broke about a week after the dairy farm spread their manure on the field adjacent to the poultry farm. Or it went from one dairy to another after they spread manure. But we don't have a lot of confirmatory evidence that could be how it's necessarily spreading. And low risk, milk trucks, migratory waterfowl, which we would have deemed a low risk because this was a single spillover until a few months ago. Now, is it really a low risk anymore? And then peri-domestic birds. So once a herd is detected as infected, a lot of times USDA's Wildlife Services goes in, they collect birds and do a lot of testing. And we see about less than 5% of the birds that they're sampling are infected. They are actually looking for infection, not evaluating those birds as fomites, which they might be a bigger risk as fomites. So I'm gonna put a big question mark on all of our low risk things now, because we've got some questions that we need to still answer. So we have been doing a lot of environmental sampling on dairies. We had I think there were 600 and some 623 samples, I believe, that we collected, and we put these into categories: milking equipment, parlor surfaces, wastewater, housing, water tanks, feeders feed, and then other samples. And really, we found about 27 to 30% of milking equipment parlor surfaces. So when we did focus on the parlor, where we know the highest concentration of viruses in the milk, that's where we found the highest concentration in the environment of those. So looking at liners, looking at drains within the parlor. We did find 14% of wastewater and lagoon samples that were positive. Most of those were in Michigan because there was a lot of concerns about it being spread through the wastewater. So the sample collections really vary by state. The onset of infection really varies by state because they had outbreaks at different times. The testing laboratories varied by state. So there's a lot of things that are different among the states and why we got positives and why we tested more samples. So when we did our mixed effects model, state really explained most of the differences that we saw. So the thing about the wastewater and lagoon, I think when we were sampling in Michigan, we tended to take samples later into the outbreak. And one of that was because we assumed that if contaminated milk was going into there, it was going to take a little bit of time before we could detect it. And then once that became a concern about spreading manure, then we went out and most of the farms were probably a month or so out from clinical signs by the time we were sampling lagoons. So 27, 29, 14 of wastewater lagoons. We did take some market samples that were all negative. So we did do some vehicle testing both in Michigan and California, looking at milk trucks, and you can see on this milk truck there's a vent on the top, and so as the truck's moving, there will be milk that comes out over the side. Technically, this is supposed to be a little milk dam up here, and then it's supposed to come down through this tube. But a lot of times those dams aren't really sealed anymore. So we see residue on the side. So that's where we kind of focused on sampling. We sampled in the cab of the truck. So the steering wheel, the pedals, the floor. We had in the Michigan instance, we had the milk truck drivers walk through a BHI foot bath for us. We sampled the tires. We sampled the back of the truck. So this is a farm load where they take the hose into the farmhouse. A lot of times they're picking up milk from multiple farms on the same load. The milk that's left in that hose could get transferred into the next farm that they visit. And then we have direct load trailers. This is one in California I hadn't seen before, but it's essentially the same kind of farm load. This hose goes into the farmhouse, but typically these are one load per truck. So we don't have them going to multiple stops on those trucks. So what did we find? Michigan, we sampled in June. Michigan broke in early April. So we're 2 months into the outbreak. It took a lot of time to coordinate that and to get the cooperative to allow us to go on and sample. We did 18 milk trucks. We took 112 samples. We had one positive detection, and that was on a direct load. This is a typical direct load where the hose is supplied by the farm. We found a positive off of this deck here on one of the direct load. High CT value of 35, so not a lot of virus there. We also found it on tires on a dead stock trailer and a livestock trailer. Both those were on the operation. I don't know that much about viruses, but it's hard for me to figure out how a virus sticks to a tire when it's going down the road at 60 miles an hour for, 30 miles, and then we swab that tire and we get virus off of it. These two were on the farm, an infected farm, when we found that. We didn't find any on the tires of the milk trucks. So California, we put a proposal in September to sample milk trucks, and we never heard anything. We thought, okay, well, we didn't get funding for that. So we heard in late March that, oh, you did get funding for that. And by that time, it had pretty much burned through the Central Valley. And I said, I think we're wasting our time to go out and sample now. Most of the farms are already cleared. And they said, well, we've got quite a few farms that we're still finding it in bulk tanks. So if you guys will come out, we'll do some testing. So we went out, we were able to, we had this in place with Michigan to get a sample of milk out of the tankers. And then the morning we started sampling, they're like, you're not touching any milk in our tankers. So we said, okay. In California, we were able to negotiate that, and they said, sure, you can get milk. Because we wanted to know if there's not any contamination in the tanker, then we wouldn't expect necessarily any contamination on the outside unless it was from a previous load. But if there was contamination in the milk, then that would be potentially a higher risk truck. So we took, we missed the first sample from the first truck of milk, but we got them from the other 19, and we had one positive detection out of the 119 samples, and it was milk from inside a tanker. So the truck was all negative, but that one, the milk in there was positive. Again, a CT of 35 off that. So I guess I left that off. But I think we just need to be careful about calling these low risk, because they're 2 to 3 months after the peak outbreak in both of these states, and we don't really know what the role of the milk truck could have been during the peak of the outbreak, when most of the farms they're hauling milk for are going to be positives. And I'll show you some CT values off positive farms. The bulk tank can get pretty low in terms of CT, so a lot more virus. So one of the things that we've kind of pushed back a little bit when this first started was we knew pretty quickly that there was a lot of virus in milk, but the thought was this is really spreading in the parlor. This is through milking practices. And as we talked to mastitis experts, they said, this is moving a lot faster than any contagious pathogen we've ever seen. And when we asked the people, they said, well, you haven't seen a viral pathogen before. And we said, well, that's true, I guess, in cattle, but there's a lot of things that have to happen just right for a virus to get up into the mammary gland and get into the tissue. And it seemed like it was moving faster than that. So we kind of focused a little bit on aerosol with some of the folks here that focus on that. But this paper came out in February, and it talks about looking at, this was poultry outbreaks in the Czech Republic, and looking at genetic data, meteorological conditions, trying to look at that. So what they did is they've got these 3 farms over here that were infected. They looked at the genetic sequences of the farms, and then where their air intakes were on these poultry farms, and how the wind direction and wind speed and landscape might have contributed to that. So their kind of suggestion was, there is a potential for aerosol transmission. We were thinking that back in 2022 when we were in Pennsylvania, and we were seeing the broiler flock that's a mile downwind from the large layer facility that's now undergoing depopulation has exactly the same sequence, and we couldn't find any connections between the layer facility and the broiler facility. We also saw that in Michigan, where we had a dairy farm that was about a half mile from an infected poultry farm. They both ended up with exactly the same sequence. And again, no connections there. The prevailing wind potentially could have moved it from the poultry farm to the dairy. So we're kind of focused on, well, aerosol might play a bigger role. And then Scott thankfully introduced me to this paper yesterday that talks about the outbreak that happened in 2015. And essentially, one of the factors associated with the early part of the outbreak was soil disruption in a nearby field within 14 days of the outbreak. So was there virus potentially in that field that then got aerosolized and then moved through the wind into the barns during that event? So when we went to California, and one of the things, we started working with a private veterinary clinic there, and they enrolled 19 farms, and they did daily bulk tank samples. And some of these farms had 6 bulk tanks that they were sampling every day. So we're 30 days into this, and all 19 farms are still negative. We've spent our budget on sampling all these bulk tanks. We're like, when is a farm going to break? So I got a call on Sunday that there was some mess up with samples, but this farm had their first positive on Thursday. So we flew out on Monday. We were on farm Tuesday, and we thought, if we can get on a farm early, because most farms we get on, it's 2 weeks after clinical signs have started, we're going to figure out where this virus is, and we're just going to know a lot more about what's going on. So when we went out there, we did a lot of sampling. This one on the left here, we're just running this through a little sampler. We'd stand in front of the cow for 5 or 10 minutes, and we were looking for cows that were either in the sick pen or had a lot of nasal discharge, because we didn't really have the typical sick cows that we see with high path AI. We put up this one. I think this might be Monsi's actually, in the parlor where the units were coming off, because we thought if that's when the units are coming off, there's going to be some milk splatter there. Maybe we would pick up some virus there. And then we talked to some other folks that said the problem is that's too low of total volume of air that you're sampling, so you need to get one of these big carrier units. We put this above the holding pen. Unfortunately, we bought 2 of those units, and then the filters became unavailable. And when you go to pull the material out of the filter, you pretty much destroy it. So now we're left with machines that don't have filters for them. Needless to say, we collected 350 samples that day. A lot of those were cows that didn't necessarily have clinical signs other than nasal discharge. We sampled everybody that had already been in the sick pen, and we came up with 0 PCR positive samples. We're like, where is this virus hiding? Ultimately, when this herd did break with clinical signs other than nasal discharge, they had no problem finding the virus on this farm. So this is the 19 farms that this private vet clinic enrolled, and BC and BO were the first farms that broke. They broke in late October. And then there's those other 17 farms, and they broke within 3 days of a wind event that happened. And as you can see, I mean, those are, maybe it's hard to tell, but Merced, Turlock, Modesto, it's a wide geographic area. So I teased the veterinarian a little bit. I said, how did you get all those farms infected within 3 days from going and picking up bulk tank samples, which he'd already been doing for 60 days. So we started working with the CSU Atmospheric Science Department and just said, can you guys go back and at least see if there's any evidence that wind could be playing a role in disease spread? Do we have favorable winds that could be moving it? So what they did is they went back, and so this graph looks 3 weeks back, and this blue dot represents a potentially infected farm. California didn't tell us exactly where the most closely related farm was, but they said it was in this county, kind of south and east of BC. So this red dot is BC, and all the lines are converging on BC based on the wind direction. So all these winds are coming out of the north and west, and we did have a little bit of winds coming out of the south and east here that were blowing toward that. That was 3 weeks prior to BC's detection. 2 weeks before, we had no winds that supported that. And then when we get a week before it, we had a lot of winds that were coming through that direction up to BC. So at least in this one, we've got some evidence that the winds could have been playing a role, but what we want to do is now we want to go get whole genome sequencing from all these herds, infection onset date, and then try to put the pieces together rather than having them kind of model every single potential. But the fact that all 17 of those farms broke within 3 days just like in Colorado, people said, it's moving with the wind. You just need to look in that. And what we think is that, as we talked today and yesterday, is that it's probably moving 10 different ways. And so we're not going to find the way it's moving. Nothing sticks out. Milk trucks aren't sticking out based on we sampled them a little bit late. The peridomestic birds aren't sticking out, but maybe we're not sampling correctly. So we're probably not going to find one thing. I can guarantee you we're not going to find one thing because we already know that they're moving through lactating cattle. The question is, how are they moving on those farms where they're not moving cattle? And the California, I don't know if you've heard the story about California, how they got infected, but they had a load of cattle, at least one load of cattle that went to Idaho to a previously infected farm. After a point in time, and I think it was like 2 to 3 weeks, the owner decided he didn't like those cattle. So he sent them back to California. And my understanding is on an expired CVI, so they weren't tested prior to moving back to California. The California dairyman that sold them didn't want them because he sold them. So he sold them to his neighbors. And so I think that shipment of cattle went to about 3 farms in California. And then this owner had to pull his kids out of school, had death threats against him for bringing high path into California. But it seems to me like maybe the Idaho dairyman might have been the one that sent them illegally. So early detection, this is the Tuesday we're out there, we're collecting samples. Before I do that, I don't know if everybody's familiar with PCR, but before I talk about results, PCR is polymerase chain reaction, and CT values are cycle threshold. Just a little background because it's kind of counterintuitive. The more virus is there, the shorter the amount of cycles we have to do to find it. So a low CT value means that there's more virus there, and a really high CT value, like a 35, like I showed on the milk trucks, means that there's really very little virus there. So it's inversely related to potentially viral load. So, but the CT values are exponential and represent the approximate amount of RNA that's amplified. So for every 3 CT decrease, there's approximately 10-fold increase in the copies of RNA in the sample. So we typically think of 40 as being 0. That's what USDA is currently using. If we get down to 33, that's 40 copies of RNA. We get down to the low teens, and we're at 40 million copies of RNA if we have 100% efficient. But this is just detection. This is looking for the RNA. It's not telling us whether that's a live virus or whether it's infectious or anything. So this was Farm BC, like I said, the first herd. 1024 was the first bulk tank detection. They started noticing this nasal discharge on 1028 in about 10 to 20% of their cows. And then we were out there on the 29th. And this is their bulk tank on the top. You can see it dips. This is kind of interesting because we in almost all the ones we look at, we see that dip where it goes down. Like we have a few cows that get infected initially, and then it's more of a propagated epidemic. Initially, I thought this was like a point source, but I think some of the things are saying maybe this is more of a propagated epidemic within the herd. And then the blue line at the bottom, dashed line, is the number of cows in the sick pen, and the axis is over here, so I'll show it after this. So this farm went from about 25 cows in the sick pen down here. By the time they were kind of at the peak, they were over 250 cows that had gotten sick. All the rest of these lines are pen samples. So once we found it in the bulk tank, then the vet clinic implemented pen sampling on 5 of the farms that we worked with. And so by the time we had clinical signs on 11-1, it was already in half of the pens on the farm. And by the time we got a week after that, it's in all the pens. And so this was a farm that was segregating sick cows into their hospital pen, as you can tell by the rise in sick cows, but we're still finding it in every pen on the farm. And if we follow that out, the pens mimic the bulk tank. It's not like it's in 1 pen or 2 pens. It's in every single pen that this farm had. And it's, like I say, it's the CT value is really mimicking what we find in the bulk tank. So we looked at that for these 6 farms. You can see that these little dips on, or little upticks on CT value on almost every one of them. This has got clinical signs. So the first report that showed this said it was a Texas herd that Iowa State and Dr. Drew Magstad looked back retrospectively. They had bulk tank samples, and they found it in the bulk tank 17 days before they saw clinical signs in the cows. In these California herds, we saw it 4 to 7 days. So these farms that was a retrospective that he did. This was, the farms knew once we found it in the bulk tank because they were on farm collecting samples. But they found it quite a bit sooner than that one herd. And most farms, PCR detection lasted about 35 days. So these are going to come back up to 40, which we're calling 0 or non-detect, about 35 to 40 days. But this farm was pretty much a flat line. And we kept collecting for, I think it was 70 days, and we still had positive bulk tank at about that level. So this is looking at some other pen samples from other farms, and we see the same trend where the pen samples mimic what's going on to the bulk tank until you get to the end, and then you can have a lot of variation, like up here, where it does then seem to settle sometimes in certain pens. This is one that's kind of chronic. The bulk tank is kind of all over. There's a lot of different pens. And I think this, in Farm BF, I think they did think that they were bringing in some new heifers, and those naive animals were the ones that were probably responsible for what they were seeing in the bulk tank based on individual pen sampling. So clinical signs, most of the cattle that have some kind of biometric sensors that the farm's using, we will find fevers in those cows for a couple days before we see a severe decrease in rumination, water intake, feed intake, or activity. These cows have a depressed attitude. The thick yellow colostrum-like milk is what we heard first when reports came out of Texas. I didn't see any of that in our cows that we sampled, but most of those were a couple weeks after the onset of clinical signs on the farm. What we saw was nasal discharge. We saw that pretty commonly. This was one of the parlors we were in, and almost every single cow, you can see 3 to 4 inches of nasal discharge on these cows, kind of a serous discharge. It did change over into a mucopurulent in these cows. We did see some black feces in some of these cows, suggesting maybe an ulcer. We did see mastitis in cows, but we really didn't get the thick yellow. That's a pretty bloody quarter there. We didn't see the thick yellow colostrum-like milk in these cows. This was a cow in Michigan the day that she was detected. So droopy ears, they just look like they feel like crap, like maybe you or I would with the flu. And she's got a little bit of serous discharge in both nostrils. We did see this cow was really pretty sick. She had more of frank blood coming out, and she was euthanized. This cow is happy, but has that bloody discharge. So it seemed to go from a serous to kind of a serosanguinous and then to a mucopurulent nasal discharge. We had some chronic cows that we tried to take in and do CT of their head and see, what's going on with some of this nasal discharge. And the equine hospital didn't want cows coming into with high path coming into the hospital. And it's like, well, we could euthanize them. And then they said, well, when we shoot them, if we're going to shoot them, then we're not really going to have a good thing to do a CT scan on anyway. So we had really good ideas, and they all kind of fell apart in terms of doing more investigation into the nasal discharge piece. This cow, this is mostly, she does have some nasal discharge over here on the left, but that's mostly saliva, and she had a big puddle of saliva that was on the feed just within a few minutes of her being locked up. Which is maybe one of the reasons why the cows are getting dehydrated, in addition to decreased water intake. So cow changes. These are 4 herds that we've looked at, looking at incidence rate. We have pretty significant differences here. Herds A, so the green, and BD, which is the one that goes up to 6.4 incidence rate per 100 cow days. They both used biometric data, which helps them find cows. I think it's a much more sensitive way of detecting cows based on rumination, feed intake, potentially temperature. And the interpretation of this then is that for this herd, 6.4, for every 100 cows in the herd, they had 6.5 cases during that day, new cases detected. So there definitely was a difference based on whether they used biometric data. The other herds were around 3 to 3.5. But we do get kind of a sawtooth appearance to some of this. And we went back and looked and said, okay, is this because they're not wanting to pull cows on weekends? Is it because the hospital pen's full? And we really couldn't find a reason that we'd see those spikes based on people looking at cows. So is this more of this group of cows is now transmitting it to other groups of cows? So the propagated epidemic part of that. We also heard early on that older cows were more susceptible. This is just one farm we looked at. So the blue represents the percent of cases in that herd in that lactation. And then the orange represents the percent of cows in that lactation in the herd. And so you can see that there's a lower percentage of cases in first lactation relative to the herd inventory. And then as we get into second and third, there tends to be more cases in those lactations. So my first thought is, why are second lactation or 4-year-old cows more likely to become infected? Does this have something to do with something that happened 4 years ago, which is now 5 years ago, which would be SARS-CoV-2 and COVID? Or is there something that's protecting the younger cattle from getting it? And I don't think we're going to get good answers on either one of those. So this is some Affy milk data from a cow that was sick. So the orange represents daily rumination, which is on the left axis. And you can see she was running about 500 minutes of rumination per day, and she drops to less than 50 minutes. Her milk production in blue, she was over 100 pounds and then dropped to less than 20 and then came back to about 70. And that's what a lot of folks saw, is that they'd come back to about 70 to 80% of their pre-disease milk production. And then the question is, what's going to happen next lactation? Because in the experimentally infected cows, they were seeing quite a bit of scar tissue in those affected udders. So this is a non-clinical cow, and oops, I think I can go back here. Unfortunately, I don't have CT values for the milk in here, but if we were taking CT values, we would expect that we're probably somewhere in the teens in terms of CT, and then this is going to go back up, within 2 to 3 weeks, she's probably going to be negative or non-detect on CT in the milk. But when we started looking at some of our non-clinical, and our clinical cows and non-clinical cows were based on what the producer presented to us. We said, we want 20 clinical cows and we want 10 non-clinical cows. So this was a non-clinical cow, again, from that farm BC, but I can show you this for almost every farm we worked on. The blue represents her milk production. She calved on 7/29. And then the orange represents the CT value of the milk samples we took. So this cow's cruising along at 100 pounds of milk, and she has a CT value of 19.3, which we would think she would be a clinical cow. But according to the producer, he didn't see any signs. There was no milk drop associated with this cow. So this kind of explains, I think, what we're seeing in Nevada, Idaho, and Arizona now, where they're finding virus in the bulk tank, but they're not necessarily seeing clinical signs. And this also kind of helps us with, why are we seeing CT values of 20 for all pens when we're segregating sick cows? Because some of the cows are shedding and we don't see any clinical signs. So I built a little calculator to try to, because it's just a dilution problem, kind of, to figure out, well, if one cow is giving 100 pounds of milk and she's got a CT of 20, what's she going to do in terms of a tanker load? So at a tanker load, one cow would pull that into the upper 20s in terms of a CT value. And if we look at this whole herd of 3,000 cows, that one cow could make the whole herd be in the low 30s in terms of CT value. So one cow out of 3,000 can cause the bulk tank to be positive. And that's just because of the exponential values of the CTs and how much virus is present in a CT of 20. So now the puzzle pieces are starting to fit together. I first thought we were working on 1,000 different puzzles because none of the pieces went together. Now we can explain, I think, in some regard, why the pen samples are mirroring the bulk tank, what we're seeing in the bulk tank in terms of a CT value of 20s when we're not seeing any sick cows in those pens. So just a few farms that we looked at. This actually was a farm we worked with in Ohio that's now published by the Cornell folks. They had 22% of their cows were sick. Farm A did use biometric data, as I mentioned. So we looked back 7 months post-clinical signs. 50% of those cows are still in the herd, represented by the blue. 2.5% are do not breed, so they're going to be gone soon. 8.5% of the cows died that were clinical cases, and we kept hearing early on that this isn't killing cows, this isn't killing cows, but it is killing cows, whether it's natural or euthanasia. There's a lot of cows that died from this, and we saw that in Michigan, where producers would tell us, well, the dead truck came today, but it was completely full. They didn't have room for our cows, so they came and picked our cows up the next day. In Colorado, I took the picture out, but we saw a dead truck driving, and it looked like there was a cow standing up on top of the dead truck. They were packed. So, and when we moved to the other side of the dairy, another dead truck showed up to pick up the cows that they had. So there are cows that are dying, and then 40% of the sick cows were sold. So they either had aborted or they dried themselves off and they weren't pregnant and were no longer productive, so they got sold. So this is impacting cattle, those sick cows, pretty significantly. We looked at 21-day pregnancy risk in this herd. And what we saw, so we know this herd brought in cattle from Texas. So we know when they were introduced, we know clinical signs. So the asterisks up there show where we see a significant decrease in pregnancy risk right after the introduction of cattle. And then we go for a couple 21-day periods where there's no difference, and then we see another difference in pregnancy risk in that herd. We also looked at abortions. So the blue line again represents 2023 numbers, 2024 is in orange. And we did see an increase in abortions out in May for this farm. Abortions are kind of sometimes a hard thing to see unless you have a visual abortion on the ground. But I can show you other farms where their 2023 abortions were higher percentage than their 2024 when they went through the outbreak. So it's somewhat variable. So we looked at milk production in this herd, and we looked at it a few different ways. We looked at peak milk, we looked at projected 305, and we looked at mature equivalent. We'd done this back in October, November, and these numbers percentages were all negative, and we're like, oh, these cows next lactation aren't doing so well. When we went back in January, and we used the days in milk cutoff, so we said, okay, well, every cow that's met 60 days in milk, and there were 244 of them, what are they doing in terms of their peak milk? They were up 6.5%, 16% in projected 305, and 8% in mature equivalent milk. The further we get into lactation, the better those estimates are going to be. So we get out to 120 days, and all those estimates are about the same. So this herd, at least, in this set of cows, didn't seem to really impact those survivors, but they're all survivors. The ones that were poor producers are already gone for the most part. So there's questions about, how does this affect overall milk production? How does it impact somatic cell count? This was the least impacted herd we could find. We used the 30 days prior to the onset of clinical signs as their baseline. And this farm had a 16% increase in somatic cell count about 3 weeks after the onset of clinical signs. And they had a 6% decrease in daily milk per cow. So this herd was, I would consider, pretty mildly affected compared to, say, this herd, where we saw a 126% increase in somatic cell count. We heard about herds that went up 300%. I just don't have the data for those. And down 15% in milk per cow. So a lot of variability, and if this is part of this is how they're managing cows, moving more cows into the hospital pen, and then that milk isn't getting into the bulk tank. So I want to go through some shedding and looking at that. So this is one of the farms in Colorado. We looked at 3 farms in California, 8 in Colorado, and 1 in Ohio. The longitudinal sampling, which you guys are planning to do as soon as you get a herd that's willing to do it. There's a lot of different cattle types that we collected samples from. But really what I want to focus on are clinical cows and non-clinical. So we picked 20 clinical. The producer picked 20 clinical. We picked 10 non-clinical. And we did nasal swabs, blood serum, and urine once a week. We did milk twice a week. And if you want to make cows happy, do nasal swabs twice a week. This is just an overall preliminary numbers. We still have some samples to get in, but 23% of our milk samples were positive, and that represented about 38% of the cattle. 3.3% of urine samples were positive, 6.5% of cattle. We saw more urine and serum than we did in nasal swabs, and the serum, PCR of serum was somewhat of an accident. We didn't know the lab was doing it until we got the results back, and then we said, "Oh, we're really glad you did that." So I want to. This is my corny tale of 2 herds. So Farm AO was the first farm we went and sampled. They had 21 clinical cows that we enrolled, and we enrolled them in the headlocks. 4 of those cows got euthanized before we got to the parlor to get milk samples from them. But we did find shedding in 7 of those, which are shown here. And then we did have 3 of the 18 non-clinical cows that we enrolled. We didn't stick to the 20 and 10 in this herd because it was our first herd. We were just figuring things out. So we didn't have a lot of shedding in either the clinical or non-clinical, but we had 89% of the clinicals seroconverted and 100% of our non-clinicals converted. And the clinical cows didn't show what we thought. We thought we would see continued shedding / detection, and then they'd stop shedding, and then we'd be done. We saw really intermittent, because each one of these, I should point this out, each row is a cow. The dates are across the top. The red is detection, and that's the CT value. 0 means we collected a sample, but we didn't detect, and then when it's blank is we missed a sample. So this was kind of unexpected. We didn't know what we were going to find in our non-clinical, but we found multiple samplings with no detection. Then we'd have 1 sampling with detection, and then they'd go negative again. So intermittent shedding or detection seems to be common in some cows. So then we go to Farm AX. We enrolled 20 and 10 like we were supposed to. We had 100% of the cows, clinical cows shedding, 19 of them in milk, which is shown here, and 1 only in a nasal swab. We had 3 of non-clinical again that shed, but their pattern looks a lot different. So we did have continued shedding, 89 days in those first 3 cows from the first positive detection to the last positive detection, and then those cows moved on to become hamburger. But the interesting thing is the non-clinical cows are now a lot different than what we saw in the previous. And the differences, one of the differences is this farm used the Future Cow udder prep, which is a wet disinfectant applied to the teats, and did not dry the teats before applying the units. So I think there's a lot more opportunity for liner slips and potential virus transmission in this herd than there was in AO. If that's, I don't know how much of a role that could have played, but when we look at 33% of the clinical cows shed in AO compared to 100% in AX, 19% of the non-clinical, and 30% in AX of the non-clinical. We didn't have any fresh cows that we detected shedding in either one of these farms. So variable farm to farm. The other thing we looked at was, I mentioned the serum, because one of the things that finding virus in serum actually proved viremia, which the experimentally infected cows, they weren't able to ever find virus in the serum. But now the finding that kind of helps us answer the question, why do we see it in urine? So all 20 cows, all these cows were in California, all 20 were positive only once. We sampled them multiple times, but they were only positive once. So milk is most commonly positive, about 60% of clinical, 18% of non-clinical. Urine and nasal swabs, not commonly, less than 10% of cattle. And then serum, at least in our kind of smaller study here, 16% of the clinical cows we found viremia in. And the sample detection duration was either one sampling on a lot of cows up to 89 days on those 3 cows that I showed you. So And the other thing is RNA detection does not always coincide with clinical signs, and that's, I think, what we're seeing in states now. So just some quick preliminary serology results from Farm AO. So I've got this broken into clinical, and then those that shed and those that didn't, and then non-clinical. And so we had 100% of the non-clinical cows, whether they were shedding or not, seroconverted at least one point during that time. And what was it? 12 out of 15 of the clinical non-shedder cows seroconverted at one point in time. So the thing that's a little bit of a mystery is why do we have cows that are positive one time on serum and then go negative again? And is that based on dose? Is that based on exposure route? We still have a lot of questions to answer there. So when we look at antibodies across these two farms, almost all enrolled cows developed serum antibodies. A little difference in the fresh cows where we had Farm AX, who I think had higher viral loads on that farm, had more fresh cows seroconvert. We didn't do dry cows on AX. 29% of dry cows on Farm AO seroconverted. The pre-weaned heifers was kind of interesting. Farm AO has a dedicated calving facility. Then they move their calves to an off-site raiser. Whereas AX, calves on farm, they leave the calves in close proximity to the cows until weaning, and then they go off-site. So is this, these were all collected about 45 days after the onset of clinical signs in the herd. So these calves are about 45 days old. Both of them were supposed to be feeding heat-treated colostrum and pasteurized waste milk, although we didn't confirm that. So is this related to a breakdown in one of those, or is this related to the calves being very close to the cows on one farm and not the other? We don't know. But did those pre-weaned heifers on AX, did they get their first vaccine? Maybe that's good, maybe not. So one of the last slides I want to show you here is we're trying to figure out, are these cows being exposed through the mammary gland first, or are they being exposed through other routes? So if you've done any somatic cell count work, there's this cool little graph that they put together that shows, previous somatic cell count, a current somatic cell count, and you look at chronic infection, new infection. So I thought, well, maybe we can do this kind of graph. So this is where they had milk and serum at the same date of collection. And down here would be they're positive on both. So the clinical are the blue cows and the orange are the non-clinical cows. And then this group up here was positive on milk but negative on serum. This group was positive on serum but negative on milk. And I don't know if you can see it. I'm biased, so I can see it really well. There's a lot more orange, or a lot more blue dots up here in the clinical cows, meaning they had milk antibodies before serum antibodies. A lot more orange dots down here, which means they had serum antibodies before they had milk antibodies. So I think that these non-clinical cows are exposed through a different route than the mammary gland, so either oral or respiratory, and developing antibodies. And that would explain, based on what we heard about in experimentally infected cows, if we expose them through oral or respiratory, they'll actually resist intramammary infection and produce antibodies. So there's our comorbidity risk. This is if you put in ChatGPT, say, draw me a picture of a cow being infected by influenza A, and make sure you show the mammary gland. So, sorry I took up a lot of time. I had to cut like 50 slides out of that to make it even close to fitting. So I'd be happy to try to answer any questions. But if you have more questions than you had coming in, then we succeeded.
Okay, we can take a few questions here. There was a question online about the federal guidelines for testing before crossing the state borders. Is that still in place, and will it continue to be in place with the current administration?
I haven't heard of any changes to the federal order coming.
So I noticed a lot of your data are CT 37, 38, 39. Were any of those or all of those confirmed with re-extraction retesting, or are these all singleton tests, and do you believe all of them?
That's a good question. So at CSU, what they do, I believe the cutoff is 36. If they have a 36 or higher, they repeat that sample. If they get a non-detect on that, they call it a non-detect. If they get detection, then they call it the lower of the CT values. We did the same thing with the serum PCR because that was kind of a hot topic at the time. So we reconfirmed those. I think there were 3 that went from the mid-30s to non-detect, but there was 2 months between testing. So we said, well, we're believing the first test on those. But if they went, if they were above 36 and they went negative, then we called them negative.
Are they using robotics or is this hand?
CSU, I'm pretty sure would be hand. I don't know about ISU. They might be using robotics.
Yeah, good question. Thank you for the presentation, Jason. How frequent is that all those dead cows, they would do necropsies on farm and look at the distribution of the virus either in the respiratory tract or, in other tissues? Is that something commonly done or not?
No, so there's very few necropsies that are done, especially at the to do this level of necropsy, you'd have to take a lot of tissues. And most of the time, if they're even gonna do necropsies, I think it's just a gross visual exam and then they move on. We have seen, like in the Ohio herd, they did some necropsies early on and they found it in a rumen wall. They found it in places you wouldn't expect to find it. We tried to implement that, but because of logistics, we couldn't get it done in Colorado. By the time we got everything in place, the cows were done dying.
There's another question on Zoom about risk to consumers and consumer milk supplies. Has there been any testing done of milk that could reach the consumer level? I'm guessing pasteurization probably takes care of that.
People took great interest in that very early on, and there were studies done that looked at pasteurization, and pasteurization was effective. I did hear some research, I think last week, that showed that when we take milk and we put virus into it for pasteurization experiments or other things, that is not the same milk as milk that comes from an infected cow. And milk from an infected cow may be a little more protective depending on what's going on with the virus and its attachments and whatever. So I don't think it's a risk, but I think we probably need to make sure and use milk from an infected cow if we can to try to continue to test that.
Don't drink your own milk. You can just say that out loud. Don't drink your own milk.
Yeah, of course.
Unless, yeah.
So Jason, one more. I couldn't help, you're finding these PCR positive samples in serum, right? About the same rate as in urine.
Higher.
And higher than, and also in nasal swabs. And we know that's not been the recommended Nuln procedure, but can you make an additional comment about looking for this virus in the serum and what that might mean?
Well, I think what it told us is that it helped explain a lot of things that were going on, because we were told that there doesn't seem to be a viremia associated with this. And then the question is the viremia coming from an intermammary infection, or is it coming from oral respiratory exposure? And I can tell you, in those 20 cows, every single one of them had it in the milk the day they had it in the serum. So my thought now is that viremia is primarily coming from an intramammary infection where we have disruption of the blood-mammary barrier due to inflammation of the mammary gland, but that's a guess.
Okay, let's thank our speaker one last time. Ride swag.
Oh, thank you.
Minnesota coffee. Nice coffee. And a cow. And a cow. Yeah, thank you so much.